So we said the resting membrane potential was pretty much what was happening in a neuron. So let's draw a neuron here. When it was at rest, right? A neuron at rest here. So we have our neuron, and we said that the inside of this cell was more negatively charged than the outside of this cell. And the specific voltage here was negative 70 millivolts, right? And this was a neuron at rest. Now, what we went on to describe was how this neuron got to that negative 70 millivolts as its resting membrane potential. Okay, so let's draw the inside of this cell. And again, keep in mind that this is a neuron at rest, just chilling out. And we said that there were sodium and potassium across the membrane here. So let's draw us. Potassium is at a higher concentration inside this cell. Let's draw those as these squares. And then sodium was at a higher concentration outside the cell here. Okay, so this is your sodium, and this is your potassium. Okay. The first thing we said, just to sort of make some notes here, was that the membrane permeability to potassium, can you see this? Yes. The membrane permeability for potassium was 25 times more than sodium. What did that mean? What did that 25 times more mean in terms of increased permeability? We spoke about this.
25 more channels.
Right, more channels, perfect. So there were more channels for potassium. Let's draw 3 of them here, versus channels for sodium. So we got 1 sodium channel compared to our 3 potassium channels, right? Now, the first thing that we have established was the concentration gradient, which we can already see here, right? We have sodium at a higher concentration outside the cell, potassium at a higher concentration inside the cell. So therefore, we have a concentration gradient. Okay, and that concentration gradient is such that potassium wants to move where? Where does potassium want to move? Outside of the cell. Let's draw that here. So this is our chemical driving force for potassium here. And sodium wants to do what? Move into the cell, right? So this is our chemical driving force for sodium. And let's just make a note. Sodium moves into the cell. Potassium moves out of the cell. Okay, so that's our concentration gradient. Now, as sodium and potassium move according to their concentration gradient, what then happens? Because we have more potassium channels than sodium channels, what's the net effect here?
The cell becomes more. Can you say that question again?
Right, so what's the net effect of sodium and potassium moving according to their concentration gradient, keeping in mind that there are more potassium channels than sodium channels?
The cell changes.
Okay, come on, you can say it. You're on the right track. Right, perfect. So the inside of the cell then becomes more negative because we have more of a positive charge leaving the cell than we have positive coming in. Again, both of these are positively charged ions, and we have a smaller, we have less doors, so to speak, for sodium to get into the cell, and more doors for sodium to get, potassium to get out of the cell. And so what we have is a net effect of more positive charge leaving than coming in, so the inside becomes more negative. Okay. What does that then bring us to? So let's make a note here. So more potassium leaves, inside becomes more negative. What does that then lead us to? Inside of the cell is more negative, outside the cell is more positive. What is that? Come on, we spoke about this. Two types of forces. One was based on concentration gradient, one was based on separation of charge. What was that second force? Right, we have an electrical driving force because of the separation of charge, which is now established. So here we have an electrical gradient. Okay, and in this electrical gradient, the inside of the cell is more negative, and these are two positive ions that we're speaking about. So what is the direction that these ions want to move in? They want to move into the cell. Perfect. So we have an electrical driving force. Let's draw that in blue. For sodium to move into the cell. Okay, this is also sodium. This is the electrical driving force. This one was the chemical driving force. Okay, just make a note here. And then potassium also wants to come into the cell. Remember, it's also a positively charged ion. And this was potassium. Okay. So we have sodium with a chemical driving force and an electrical driving force in the same direction. That is going to magnify or amplify, the strength of that driving force. So sodium really wants to get into the cell. Potassium, on the other hand, has a pull. It has a chemical driving force in one direction and a electrical driving force in the opposite direction. So there's not as strong a magnitude for potassium to move into the cell. Move out of the cell. Okay, so the net direction here is that potassium will leave the cell, but not with the magnitude or the strength that sodium is entering the cell. All right, let's make a note of that. So both ions move into the cell. All right, this is according to the electrical gradient. And the net here, right, the net movement is that sodium rushes into the cell, whereas potassium is going to trickle out of the cell. Out of the, all right, so everyone understand what I mean? Sodium really wants to get into the cell because we're combining the same direction of these forces, electrical force and the chemical force. Potassium, on the other hand, is moving out of the cell, but it's trickling. There's not as strong a magnitude in terms of this force. And again, this is the electrochemical driving force when we combine these two forces. Alrighty? And so the fifth step here, or the final part of establishing the resting membrane potential, is when both of these ions move according to their gradients, right, their electrochemical gradients, also bearing in mind the increased permeability for potassium versus sodium. What we establish is a membrane potential at negative 70 millivolts. And this is steady state. Steady state means that this is the membrane potential where both these ions sort of balance each other out. We don't have any more net movement of ions at this. And this is when the neuron is at rest and it's at steady state. Okay. We also want to bear in mind the presence of a specific protein in the membrane of the cell. What's this protein that's responsible for establishing these gradients? What's this protein called? Anyone? So we got leak channels that are moving ions in and out, but what protein is establishing the gradients of sodium and potassium? The pump, right? The sodium potassium ATPase pump. And again, it's going to pump 2 potassium into the cell and 3 sodiums out of the cell. Okay, so just to reiterate here, the sodium potassium ATP pump is pumping 2 potassiums into the cell. They are then going to leak according to their concentration gradient out of the cell. We're pumping 3 sodiums out of the cell, and sodium is going to go on the other end and leak right back according to its concentration gradient. So the pump is vital, crucial in establishing the initial concentration gradients across which ions can then move and generate that resting membrane potential.